Hello! Today I will be talking about this hypothetical developmental network where the pathway leads to the spike formation in certain cells of the ectoderm in a beetle. This will help explain why not all the cells in the ectoderm have spikes. So here's the network of normal strains with the different genes that either repress or activate transcription of each other to eventually lead to the formation of spikes. The arrows represent activation and the dashed lines represent repression. Below there are mutant strains listed, and we can use the developmental network to see if the cells will still be able to produce spikes despite the mutations in some of the genes. First, we have a mutant allele in place of P. Without the P gene product, the I gene cannot be produced, so the inhibition of K does not occur, leading to the repression of E, and the spikes are not produced. Now we have a mutant in the I allele. Without the I gene product, it cannot inhibit K, which inhibits E and again stops the spike production. Now there is an absence of both I and E gene products. We already know what would happen if the I gene product was not present, but if E is not present, then spikes are still not produced because there is no E gene product to activate the spike phenotype. Lastly, there are mutants for the S and K alleles. Without S, the P gene cannot be activated, which activates I, but K does not repress E because it is absent. Therefore, we finally have spike production due to E gene product activation. Since multiple gene products influence the transcription of the gene for the spikes phenotype in the beetle, sometimes spikes are not formed in every cell. Possibilities for a decrease in transcription of the particular genes could be histone methylation, DNA methylation, or chromatin remodeling. Mutations could also be caused by factors in the environment, such as epigenetic changes, which can change the phenotype of a cell or organism without changing the genotype. Thank you so much.